Hello friends and welcome. Today we are going to talk about Spheroplast. The main objectives of today's deliberation are: number 1, to give basic idea about Spheroplast. Second, to find out general characteristics of Spheroplast. Third, to know about the protocol of Spheroplast formation. And finally, to give a detailed insight about applications of Spheroplast. To begin with, let me give a brief introduction. A spheroplast is a cell from which the cell wall has been almost completely removed, as by the action of penicillin. The name stems from the fact that after a microbe's cell wall is digested, membrane tension causes the cell to acquire a characteristic spherical shape. Spheroplasts are osmotically fragile and will lyse if transferred to a hypotonic solution. Spheroplasts refer to the spherical shape assumed by gram-negative bacteria upon partial or complete removal of the cell wall. Cells with compromised cell walls assume a characteristic spherical shape to better withstand the rigors of its surrounding environment. They are also extremely sensitive to osmotic and mechanical shock. Spheroplasts have two, an inner membrane and an outer membrane. The peptidoglycan is the main stress-bearing layer of the bacterial cell wall, and the peptidoglycan also gives the bacterium its shape. In the laboratory, weakening the peptidoglycan network in the cell wall generates both protoplasts and spheroplasts. By exposing bacteria to an enzyme called lysozyme, the interconnect strands of the two particular sugars that form the peptidoglycan can be cut. When this is done, the peptidoglycan loses the ability to serve as a mechanical means of support. Now, let us discuss general characteristics of spheroplasts. Number 1, a bacterial cell from which the rigid cell wall has been incompletely removed. The bacterium loses its characteristic shape and becomes round. Second, the spheroplasts can adjust their internal osmolality by increasing their volumes more than 3 times upon osmotic downshift. Third, at constant external osmolality, unfolding and refolding of the membranes are reversible, thus providing an apparent elasticity of stretching. Number 4, until the spheroplasts are swollen to their volume limit, their membranes are tensionless. Number 5, the membranes cease to refold when spheroplasts lose viability, implying that the membrane reservoir is metabolically maintained. The area of the spheroplast membrane changes to a new equilibrium value by a relaxation process, and the relaxation is loading rate dependent. The equilibrium tension area relation is reversible. Now, let's talk about different protocols for preparation of spheroplasts. Number 1, treatment with EDTA and lysozyme. This method was given by Voss in 1964. In this method, a nutrient broth of 100 milliliters in a 500 milliliter flask is inoculated from a 16 hours agar culture of Escherichia coli and shaken for 24 hours at 37 degrees Celsius. 4 milliliters of the culture are centrifuged at 20 degrees Celsius and bacteria washed twice with sterile distilled water and resuspended in 2 to 5 milliliters of water. 2 milliliters of the suspension are added to 0.3 milliliters EDTA solution of 0.03 molarity, 0.24 milliliter lysozyme solution of 500 microgram per milliliters, 0.2 milliliter tris buffer of 1 molarity, 1.2 milliliter sucrose solution of 1.5 molarity and distilled water 2.06 milliliters and incubated at 37 degrees Celsius for 30 minutes without shaking. About 95% of the organisms are converted into spherical bodies after 5-minute treatment with EDTA lysozyme. Number 2, treatment with benzyl penicillin. It's a method similar to that of Hirokawa 1962. In this nutrient broth of 100 milliliters contained in a 500 milliliters conical flask is inoculated with a loop full of Escherichia coli taken from a nutrient agar slope culture and incubated at 37 degrees Celsius with shaking for 18 hours. The culture is then diluted fivefold with Hg broth containing 1 mg benzyl penicillin per ml. 100 ml of this suspension are shaken at 37 degrees Celsius in a 500 ml flask for 4 hours and then stored without shaking at 5 degrees Celsius for 16 hours. The suspension is centrifuged at 1500 G for 20 minutes at 20 degrees Celsius and deposited spheroplast are resuspended in Hg broth. To inactivate the penicillin, 0.1 ml of a preparation of Bacillus subtilis penicillinase is then added to 10 ml of the suspension, which is maintained at 20 degrees Celsius for 60 minutes. The inactivation of benzyl penicillin in the suspension is confirmed by an agar diffusion assay using Staphylococcus aureus dakin as the assay organism. Finally, the spheroplast suspension is centrifuged at 1500 G for 20 minutes at 20 degrees Celsius and resuspended in fresh Hg broth. Number 3, treatment with EDTA. A method similar to that described by Levy 1965b is used. In this, a 16 hours culture in nutrient broth is centrifuged at 1500 G for 20 minutes at 20 degrees Celsius. The deposit washed twice with 0.1 molar tris buffer at pH 8 and resuspended in fresh tris buffer. The suspension is then adjusted with tris buffer to contain approximately 2 into 10 raised power 9 bacteria per ml. Samples of 9 ml of this suspension are warmed to 37 degrees Celsius and 1 ml EDTA solution 2 millimolar in tris buffer and pH 8 added. To a control suspension, 1 ml tris buffer only is added. Finally, let us discuss various applications of spheroplast. Number 1. Antibiotic discovery. Second, patch clamping. Third, cell lysis. And fourth, transfection. Now, let us discuss them one by one. Number 1, antibiotic discovery. From the 1960s into the 1990s, Merck & Co. used a spheroplast screen as a primary method for discovery of antibiotics that inhibit cell wall biosynthesis. In this screen devised by Eugene Duanley, growing bacteria were exposed to test substances under hypertonic conditions. Inhibitors of cell wall synthesis caused growing bacteria to form spheroplasts. This screen enabled the discovery of fosfomycin, cefamycin C, thinamycin, and several carbapenems. Number 2, patch clamping. Specially prepared giant spheroplasts of gram-negative bacteria can be used to study the function of bacterial ion channels through a technique called patch clamp, which was originally devised for characterizing the behavior of neurons and other excitable cells. To prepare giant spheroplasts, bacteria are grown in a medium containing chemicals that prevent the cells from dividing completely. This causes bacteria to form long snakes that share a single membrane and cytoplasm. After a period of time, the cell walls of the snakes are digested and the bacteria collapses into very large spheres surrounded by a single lipid bilayer. The membrane can then be analyzed on a patch clamp apparatus to determine the phenotype of the ion channels embedded in it. It is also common to overexpress a particular channel to amplify its effect and make it easier to characterize. The technique of patch clamping giant E. coli spheroplasts have been used extensively for studying the native mechanosensitive channels of E. coli since 1987. Recently, it has been extended to study other heterologously expressed ion channels, and it has been shown that the giant E. coli spheroplast can be used as an ion channel expression system comparable to the Xenopus oocyte. Number 3, cell lysis. Yeast cells are normally protected by a thick cell wall, which makes extraction of cellular proteins difficult. Enzymatic digestion of the cell wall with zymolase creating spheroplasts renders the cells vulnerable to easy lysis with detergents or rapid osmolar pressure changes. Gram-negative bacteria such as E. coli are subjected to outer cell lysis by osmotic shock and the action of enzyme lysozyme and ethylene diamine tetraacetate, that is EDTA, the chelating agent, with subsequent release of periplasmic gap content and spheroplasts containing inner cell content into the sucrose medium. Lysozyme catalyzes the hydrolysis of the peptidoglycan layer, while EDTA destroys the outer membrane, facilitating enzymes access to the inner layers of the cell wall. The periplasmic enzymes released during the rupture of E. coli outer cell wall include an alkaline phosphatase, a cyclic phosphodiesterase, an acid phosphatase, and a 5' nucleotidase. Sucrose tris-hydroxymethylamminomethane HCl medium is essential to the preservation of the integrity of the enzymes upon lysis. EDTA aids cell hydrolysis by binding to divalent ions such as calcium dipositive and removing them from the wall, thus softening the wall for further lysozyme action. Number 4. Transfection. Bacterial spheroplast with suitable recombinant DNA inserted into it can be used to transfect animal cells. Spheroplasts with recombinant DNA are introduced into the medium containing animal cells and are fused by polyethylene glycol, that is PEG. With this methodology, nearly 100% of the animal cells may take up the foreign DNA. Upon conducting experiments following a modified Hanahan protocol using calcium chloride in E. coli, it was determined that spheroplasts may be able to transform at 4.9 into 10⁻⁻⁴. With this, we conclude today's lecture. Hope this was helpful. Thank you and goodbye.